Hi, everyone. I'm excited to welcome you to episode 716 of the People Behind the Science podcast. I'm your host, Dr. Marie McNeely, and today we are resharing our interview with our guest, Dr. Dominic D'Agostino, originally published on May 29th of 2017. Listeners, Dom and his team develop and evaluate metabolic-based therapies to treat cancer and other diseases, and they investigate the physiological, cellular, molecular, and epigenetic mechanisms of these different therapies to better understand how the therapies work and how they can be optimized. And in our interview, Dom shares some fantastic stories from his life and research. So get ready for this episode of People Behind the Science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Today, our guest is Dr. Dominic D'Agostino. So, Dom, welcome to the show today. How are you?
I'm doing great. Thanks for having me.
Excellent. Well, we are delighted to speak with you today and learn more about your life and your work. But before we dive into the details, I do want to give our listeners a little bit of background here. So, listeners, Dom is an associate professor in the Department of Molecular Pharmacology and Physiology at the University of South Florida. He's also a research scientist at the Institute for Human and Machine Cognition. Now, Dom received his bachelor's in nutritional sciences and biological sciences, as well as his Ph.D. in neuroscience and physiology at Rutgers University. Afterwards, Dom conducted postdoctoral research at Wright State University and at the University of South Florida before joining the faculty there at USF. And Dom is here today, listeners, to talk to us about his science and his life. We want to hear about it all. So, Dom, can you start by telling us what you like to do with your time when you're not very busy doing science?
Well, I like to get out in nature. My wife and I are really big in being outdoors and as much as possible, being at the beach. And last week we were celebrating our one-year wedding anniversary, and we did a pretty extensive hiking, and we like to do kayaking, mountain biking, horseback riding. But one of the things that we probably put a lot of our time into, and it's somewhat work-related, so maybe I'm cheating here, is scuba diving, doing field biology research. So my wife is a marine biologist, and she gets to study manta rays and the cognitive and behavioral aspects of manta rays. But to do that, you have to go to places like Indonesia and Fiji and Palau and all these crazy places. So when I can take time off my schedule at the university, I go with her, and it's a vacation, it's our downtime, but at the same time, we're collecting lots of video footage, DNA, and information from manta rays at all these awesome dive sites. So that takes up a big chunk of our free time when we're not quote unquote working. It just doesn't feel like work, but we're still collecting data and publishing from our vacation experiences, I guess you could say.
That is fantastic. So what is your favorite place to dive?
I would have to say we've been all over, but I think Hawaii has been one of the better places. We were in Philippines for our honeymoon last year and diving with thrasher sharks. It was a very remote location off of Malapascua, Philippines. So I would say that place, or in Kona, Hawaii, off the coast there are population of manta rays where you can go every night and have 100% chance of seeing them. So we tend to do a lot of diving there just because we can collect more data when we do that. And it's just a thrilling experience to dive with these creatures that are the size of your car and to get so close to them that they're touching you and doing these barrel rolls. And they are the fish with the largest brain of all fish in the ocean. And they have very unique cognitive and sensory abilities. So you actually feel like you're with them under there. And it's one of the only places in the world where you can do that. So I'd say that's probably the highest on my list.
Well, that sounds phenomenal. I've done a tiny bit of scuba diving myself, so I'm going to have to make it out there.
Yeah.
So great to hear more about your life outside of science and the little bit of science, I think, that permeates it as well. But I do want to talk more about your research specifically and your scientific side. So Dom, can you tell us, what do you tell people when they ask you that question? What do you do for work?
Well, I kind of figure out where they're coming from and take it from there for a description. My work as a professor, like in a tenure track position, was really focused on studying the neuroscience and the physiology of extreme environments, how it relates specifically to the undersea environment and also the altitude or the space environment. So most of the funding that I've acquired through my tenureship at the college has been through the Department of Defense and the Office of Navy Research, in particular their undersea medicine program. And we developed various technologies, for example, like a microscope inside of a chamber, and that chamber can be pressurized or decompressed to simulate a particular environment that, for example, a Navy SEAL will experience or an astronaut will experience. And then with the microscope inside there or electrophysiology equipment, we do like patch clamp electrophysiology or intracellular recordings or even telemetry equipment if we're doing whole animals, we can simulate those extreme environments, whether it be 20 atmospheres of hyperbaric pressure in the undersea environment or 5 atmospheres, or we can pull a vacuum on the chamber and simulate the top of Mount Everest. And inside the chamber, we can measure brain activity, EEG, diaphragmatic EMG, heart rate activity, or with some technologies, we can look down at the level of the mitochondria and measure mitochondrial reactive oxygen species production or membrane viscoelasticity if we're using the confocal microscope or the atomic force microscope in that environment. So it's really about studying our cellular, molecular, and physiological processes in these extreme environments. And that's kind of my niche. So our lab has this unique equipment that allows us to do that. And that became our primary focus. And then it led to things like our cancer research program and other neurological diseases that we study from the observations we made in that type of equipment.
Well, this is really cool. And are you to the point of doing these types of experiments in human participants, or are you kind of working more at the cell and model organism level?
We're right at that point right now, actually. We have 5 IRB protocols being reviewed right now by several institutions, including NASA, where we will be studying some of these things in humans. And some of the things that we plan to look at humans exposed to these extreme environments is things like metabolomic profile, looking at blood metabolites and getting more or less a metabolic fingerprint of that person's metabolic physiology in that extreme environment, whether it be hypoxia. We're doing some work on Kilimanjaro and then or the undersea environment with NASA's NEMO mission, which is NASA's Extreme Environment Mission Operations. And just recently, I was selected as a crew member to work with astronauts and planetary biologists in this environment. And it's NASA's Earth-based space analog that can simulate environments and mimic environments that have relevance to a deep space mission, like to being on Mars. And various technologies are studied in these Earth analogs and kind of vetted out more or less before they go to space. And I guess about 70 to 80, maybe as much as 90% of the astronauts need to go through this training. And I'm in academia and not at NASA, so I'm not an astronaut in training, but I'll be working with them as a scientist to study some of these things. And we're looking at autonomic regulation, like heart rate variability. We're looking at the microbiome, like how does the microbiome change in these extreme environments? Body composition, because loss of muscle and bone is something to consider. Psychological things like teen cognition. We use the NIH toolbox for that, and my main focus is metabolism. So yeah, we are just at that point where we're translating our basic molecular and cellular findings into human studies.
Well, you have so many exciting projects going on in the lab right now. I think we could chat all day about them, but I do want to talk a little bit about what motivates you. I think as a scientist, there's so much to get done in the lab, let alone so much to get done in life in general, that I am always looking for things to motivate me and keep me going. So, Dom, do you have a favorite quote or a saying or a force that really motivates you?
Motivation, yeah. I mean, I draw it from so many different areas of my life, but I think everybody in scientists, and you may agree, are very curious. And it's that curiosity and thinking about the big picture questions and working realistically in science using the scientific method. We can do things that can move us incrementally towards that understanding. For example, how the brain works. And also, when we're advancing new ideas, I think one of the most motivational quotes would be to avoid criticism, say nothing, do nothing, and be nothing, right? And we study some out-of-the-box ideas, like using certain dietary approaches for the metabolic management of cancer, for example, because we study the ketogenic diet and ketone supplementation. And there's a backlash from some aspects of general scientific community against this until a secure foundation of data is there. When we first started studying this, there was no registered trials on clinicaltrials.gov, but now there's about 20, I believe. But because I was studying things that you typically wouldn't face criticism for, but when we started studying some out-of-the-box ideas, it was kind of new to me as a scientist to get some criticisms. And I think maybe sometimes we can take things too personally, but I think as a scientist, you're always going to be competing because the pot of money is pretty small, and other people will have conflicting views or even conflicting data from your own. So it's really important to be able to accept criticism and view things from the other person's perspective.
Definitely. And I think sometimes the more criticism you get, the more indication that you're onto something interesting. At least the direction of your research is going that way, because if no one's saying anything, if no one's complaining, then maybe it's a sign that they're not interested in your questions. So I think you've stumbled on something interesting.
Yeah, that's probably a bad thing. Whenever you're kind of studying paradigm shifting ideas, you're going to face criticism. And I think it's really important, and I've had to work on this myself, is that your ideas, when you have them and you collect data, to change your ideas along the way, according to not only the data you're collecting, but other people's data too, and incorporating that data into whatever model system that you're studying to avoid bias. So you really need to consider and carefully consider the criticisms and critiques that other people have in your work, and to incorporate that not only in your experimental design, but even in the discussion of your results, which is not always easy to do sometimes. If your grant is pretty much dependent upon testing this particular hypothesis, you have to be flexible along the way and adapt.
Absolutely. And just for any listeners out there who may not be familiar with it, can you just give a brief overview of what the ketogenic diet is?
Sure. The ketogenic diet has been around for at least a century, and it was originally used for epilepsy that was of a severe nature, what we would call grand mal seizures, or now called atonic-clonic seizures. And it really was the standard of care for epilepsy back in the 1920s, 30s, and 40s, until the advent of anti-epileptic drugs in the 50s, 60s, and 70s. And then the anti-epileptic drugs did not work that well, so there was a resurgence of using the ketogenic diet. And essentially, the ketogenic diet mimics many aspects of therapeutic fasting, and it forces your body to shift away from carbohydrate, glucose-based metabolism to fatty acid and ketone metabolism. And of particular relevance is that the brain has the metabolic flexibility to adapt from using glucose to using ketone bodies as a source of energy. And when your brain is being fueled off ketones, it's, for reasons we don't fully understand yet, it restores brain energy homeostasis in a way that can be a powerful metabolic-based approach for managing epilepsy and dozens of different seizure disorders. So we can utilize the ketogenic diet as a mitigation strategy for preventing seizures from oxygen toxicity that, for example, a Navy SEAL would get using a closed-circuit rebreather. And we can develop the ketogenic diet as a macronutrient ratio that's very high in fat, from 90 to 65% fat, depending on the type of the ketogenic diet. It has low to moderate protein and very minimal restricted amount of carbohydrates. And when you eat this, it suppresses the hormone insulin. And by suppressing the hormone insulin, it more or less stimulates fat oxidation in peripheral tissues and, most importantly, your liver. So beta oxidation of fatty acids in the liver generates acetyl-CoA, and the acetyl-CoA essentially condenses into these ketone bodies. And the liver produces ketone bodies, but interestingly does not use ketone bodies as a source of energy. So they get into the bloodstream and they provide a source of energy for for your heart and, I guess most importantly, your brain. Because if you didn't make these ketone bodies in a state of starvation, you would quickly waste away by chewing up all your skeletal muscle for glucose. And when we get into fasting ketosis, that's the benefit of ketone bodies. They're very protein sparing, and they prevent muscle breakdown, and they keep a steady flow of energy to the brain in the absence of glucose availability. And the same thing essentially is mimicked by the ketogenic diet. And it just needs to be very carefully calculated and implemented by a medical trained person, especially for a disease process. But we develop forms of supplementation that can be consumed orally that mimics the ketogenic diet and can circumvent the dietary restriction that's typically associated with achieving that physiological state. So by consuming a supplement, you can elevate the ketone bodies in certain ratios that can be therapeutic, neuroprotective, and even anti-cancer in some forms of cancer. So this is kind of like a new, maybe some people call it a disruptive technology that is emerging. And we're studying it not only for seizures, but for a number of other applications because it represents an alternative form of energy.
Certainly. Well, this is very interesting. And you had mentioned the similarities between some of the metabolics between the ketogenic diet and fasting. And I understand you have what I almost perceive as a superpower of you could go a whole day without eating and still be performing at a high level. And I'm the opposite. I'm one of these people who operates wandering through the world with a purse full of granola bars and I'm starving for lunch at 10 a.m. So can you tell us a little bit about this and how it works for you?
So the ketogenic diet is a big cornerstone of the research that I do in developing different forms of supplementation. So one of the benefits of the ketogenic diet is that by forcing your body to be fueled off fats and ketones, and it takes probably, some people say it takes 2 or 3 weeks for your body to adapt to the ketogenic diet, but I think it's more like 4 to 6 months. And it's a slow process, and some people can adapt a lot quicker than others. But when you're eating a ketogenic diet, and say, for example, you eat breakfast, and 8 to 10 or 12 hours later, you haven't eaten, your fat stores are much more accessible to your body's energy system than they would be otherwise through the suppression of the hormone insulin. And that allows a greater mobilization of fat from fat stores to be used as energy. And then the fat can also be broken down into ketone bodies. And if your ketone levels, probably most importantly here from your perspective, if your ketone levels are elevated in your blood, that makes you fairly resilient against the subjective and physiological effects of hypoglycemia. So if you get hungry and you're eating carbohydrates, your body is essentially expecting another carbohydrate meal to sustain your blood glucose levels. On a ketogenic diet, when you eat it, the hormone insulin stays relatively stable and blood glucose even stays relatively stable, but the ketones are elevated. So if you do go hypoglycemic, if you're calorie restricted, because your ketones are elevated, your brain does not perceive it as an energetic crisis. Whereas if you're eating carbohydrate meals throughout the day.
A purse full of granola bars, right?
Yeah. So your glucose goes up, and then you have a pulse of insulin that kind of goes up, and insulin's role is to facilitate glucose disposal in peripheral tissues. For example, if you eat a granola and do a little bit of physical activity, the insulin will kind of translocate the GLUT4 transporter from skeletal muscle to the surface, and you suck up glucose from your bloodstream, and your brain perceives this as an energetic crisis, more or less, and then you'll get hungry. It'll trigger cravings for you to go eat more sugar. And that kind of feed-forward process as it relates to eating and being hungry with carbohydrate meals is essentially abolished or greatly attenuated when you're eating a ketogenic diet, because you eat the meal, your fat gets elevated. You do have more fat probably going into your adipose tissue, but you also have much more fat being released. So you have this kind of fat flux in and out of your adipose tissue, and then you have the beta oxidation of fats also in the liver. And most importantly, your glucose levels, if you were to wear like a continuous glucose monitor, like a Dexcom, which my type 1 diabetic student wears a Dexcom, and there's a huge difference between if he eats a carbohydrate carbohydrate-based meal pattern, and when he shifts to a ketogenic diet meal pattern, the postprandial excursions in blood glucose are almost completely abolished. And if you look over 7 days of his glucose readings, it's almost like a very tight plot on the graph, whereas if he's eating carbohydrates, you see these lines all over the place, where sometimes he'll get a hypoglycemic dip, and sometimes it'll shoot up to 300-400 milligrams per deciliter, which could be sort of dangerous, especially if that's occurring day in and day out chronically over time. And most importantly, from a type 1 diabetic perspective, is that he can use 20% of the insulin that he typically uses on a high-carb diet. And whenever you can use less insulin as a type 1 diabetic, that's a very good thing. And if you relate this to a healthy individual, whenever they're releasing less insulin or using less insulin, that will increase their insulin sensitivity in a way. And that's a good thing. But the diet, to get health benefits on the diet generally, it doesn't have to be a ketogenic diet, but it can be restricted in carbohydrates to a degree where you're just burning more fat for fuel and you have less glucose fluctuations. And then by just getting less glucose fluctuations, it may be able to control your appetite a little bit better. And some people can do this with a quote-unquote paleo diet or low-carb paleo diet or whatever you want to call it. But I think it kind of comes down to carbohydrate restriction and then adding in more healthy fats and maybe even more protein depending on what diet you're coming from.
Gotcha. Well, Dominic, let's chat next a little bit about the adventures that got you to where you are today. I'm curious, were there people along your journey who really motivated or inspired you to become the scientist that you are today?
I'd probably have to go back to my undergraduate advisor and mentor that later became my Ph.D. advisor, Dr. Gigi Neubauer, and she was at Rutgers University, also called the University of Medicine Dentistry of New Jersey / Robert Wood Johnson Medical School.
It's a mouthful.
It's probably got the longest name of any school. Yeah, so I always abbreviated it. I started doing research as an undergraduate, to be honest, not because I was super interested in research, but as something that I could put on my CV for medical school. So it looks good if you're an undergrad. Mm-hmm. And my undergrad was in nutrition science. And then people kind of said, well, you should probably also double major in biology if you want to go into medicine. So I did that. And in addition, I worked kind of hard to get a research position to do some undergraduate research. And that kind of facilitated my interest in basic science research. And through the process of doing undergraduate research, I realized that it may be a better option for me, for my personality. I liked having the intellectual independence to do whatever I wanted, and I saw PhD scientists doing that, whereas in med school, you really have to adhere to a pretty strict regimen.
Right. There's less freedom. Yeah.
Yeah, a lot less freedom. And we work really hard and long hours in academia, but we do have that freedom, and that's very valuable to me. That really stimulated me. My undergrad mentor, which later became my PhD advisor, and I studied the neural control of autonomic regulation. She was a PhD working in a clinical department of pulmonary critical care among many different medical doctors. And she was like the thought leader of the group when there would be grand rounds. And it was very clear that she was highly respected, one of the most respected in the entire department. And in her position, she was able to facilitate some really great research projects. So she kind of convinced me that saying maybe you might want to look at a career in research. And she was instrumental in basically encouraging me to apply to the PhD program in neuroscience and physiology, and also got me into scuba diving. So she was an avid scuba diver and got me to do my training in scuba diving, which got me interested in kind of military diving as it relates to some of the problems that are experienced physiologically with the Navy SEALs. And that steered me into doing a postdoctoral fellowship project to study the cellular and molecular mechanisms of these extreme environments. So one thing led to another, which always kind of happens, and my career path got trimmed down and focused on a very specific subject, which it may seem esoteric to some people, studying neuroscience from an extreme environment perspective, from high pressure, high oxygen perspective. So hypoxia is more common, but nowhere on Earth are we ever subjected to hyperoxia unless we're in some kind of artificial environment, whether it be undersea environment or space environment.
Right.
But it was that, or we're getting hyperbaric oxygen therapy there's like 15 different FDA approved applications. So the research that I do for the undersea medicine has direct implications for understanding hyperbaric oxygen as a therapy and its clinical utility there. So I work kind of closely with that group of people too.
Very interesting. And now you mentioned that you had this research experience kind of starting in undergraduate. Did you have any interest in the sciences before that point? Or how did you start getting interested in science?
I think it was going back to senior year in my high school, and I was not a very good student, to be honest, but I was challenged by, I guess, my academic advisor at the time to take an honors biology course. And when I took that course as a senior in high school, I was also into fitness and working out and understanding the human body. And this course represented to me a way that I could better understand my body and nutrition to do better in football, which was a sport I was playing at the time, and to get bigger and stronger. So it was sort of for personal kind of selfish reasons. I became completely engrossed into biology. And it was actually a Bio 2 honors course where we do dissections of animals and have pretty high level science that we can apply to our undergraduate studies. And it was really at that point in time that I just became completely fascinated with physiology and with medicine. It's kind of sparked my interest. And then that interest grew throughout my undergrad, and then really got on fire when I started my Ph.D. program. I was reading several books a week on different subjects of neuroscience and the brain. And the 90s was the decade of the brain. So I was really excited to be studying neuroscience at that time, and still am.
Excellent. Well, great to hear a little bit more about how you got to where you are today. And we've chatted just a little bit about some of the research that you're doing in some of these different areas. Is there a particular project right now that you have ongoing, Dom, that you want to dive into some more detail and tell us about?
Yeah, we have a lot of projects. So several PhD students in my lab, and one of them studies cancer cachexia, so muscle wasting as it pertains to cancer. And we are looking at various metabolic pathways that can mitigate the muscle loss and wasting, with the idea that you can significantly extend the quality of life with that. So that's one project I'm really excited about. And I guess the other project that's kind of ongoing now, or at least we're setting up to do it, would be the NASA NEMO 22 project. And it's so exciting to me because we're able to translate some of the research that we've been working on for over 10 years into human subjects, and in particular, very interesting human subjects, which would be astronauts that are going to be exposed to extreme environments. And that project is specifically looking for when you're in a saturation environment under the water, well, we'll be breathing air in a hyperbaric environment. So if we shot to the surface, we would die because the gas would try to come out of our bloodstream and create bubbles. So the environment represents some things analogous to space, where you can't leave that habitat. You have to stay in that habitat. And then you have a list of tasks to do that are highly relevant to things that would be done in space. And at the same time, we'll be able to collect data on metabolism, on how energy metabolites change, especially in response to a ketogenic diet eating regimen, how stress indicators will change. We can look at heart rate variability, which gives us some insight into the sympathetic versus parasympathetic nervous system and how that changes, especially in regards to sleep. Because during our normal activities that we're doing in the extreme environment, my sympathetic nervous system will be all over the place. But when we sleep, the heart rate variability that we can collect during sleep is more normalized, I guess you could say, and will be a good reflection of the overall stress that our system is under. We're also interested in looking at body composition alterations and how a loss of lean body mass may be occurring in this extreme environment and ways that we can potentially mitigate it, even with ketones, because I talked about ketones being an essential metabolite responsible for anti-catabolic or protein-sparing effects during fasting. So we wanted to determine whether elevating ketone bodies can also serve an anti-catabolic role in extreme environments. And then we have a battery of psychological tests. We'll also be doing the NIH toolbox, which looks at a variety of things. So I'm really excited about that. And we've been in the process for several months in setting all that up and doing the training necessary to conduct that mission. So that's the thing that's on our task list right now.
Dom, I love it. This sounds like a really exciting project, and it sounds like you're at a really exciting point now being able to translate it into humans. And we've talked about a lot of the fantastic things that you're doing right now in your laboratory and also some of the things that you've been able to do throughout your training to get you here today. But we haven't really talked about the challenges, the struggles, the downtimes. And I know every scientist goes through these tough times. So I think it's really important to share these stories with our listeners to give them a realistic view of the career path and life of a scientist. So do you have a story of your own of a failure, a struggle, or a challenge that you can share with our listeners today?
Many along the way, actually. When you're in science and academia, there's always politics that you have to deal with too in your department, and even politics associated with getting funding. So there's certain types of research that are not viewed in the eyes of government as something that should be funded. And nutrition is probably one of them. It's kind of marginalized as something that may not have a big impact on health and wellness and treatment of diseases. But we think of nutrition as the cornerstone of health and can be used as a powerful metabolic therapy for many different disorders, and most importantly, probably prevention. So as a career scientist in a college of medicine, it is of utmost interest to the department and the university as a whole to be funded by the National Institutes of Health, the NIH. NIH R01 grants, it should be sustaining it. These grants are really hard to get for the things that we study, nutrition protocols. But the Department of Defense and the Office of Navy Research and organizations like NASA have been much more welcoming to funding some of the projects that we do. So for me, staying afloat as a scientist, especially through a tenure track position, I would have completely failed if I would have focused specifically on the National Institutes of Health funding. So I had to reach out and kind of be resourceful in some ways. So I've repeatedly failed over and over trying to get funding from the common funding path that a scientist in my position would be going after. And I had to do some reaching out to not only military funding sources, but also private industry to fund some of the things that we're doing, especially in the realm of cancer research. So that's sort of the major obstacle that I've had as a scientist, is that I had to really focus my efforts on different funding paths than what's traditionally done in someone in my department. And if I take a step back even in graduate school, as far as problems are concerned, it was extremely important to be persistent. The technique that I focused on as an undergrad and a Ph.D. student was called patch clamp electrophysiology. And it was very difficult to do this on the types of cells and neurons that I was studying. And it literally took me about 2 years to get my first usable data that I could put into publications.
Oh, wow.
It was, I think, probably at least a dozen times where I was just going to give up and take a different career path. But I was told it's common for this type of technique, doing whole cell perforated patch clamp technique, where you have an electrode and you bring the electrode down and you attach it onto a neuron, whether it's in a brain slice or in a cell culture preparation, and then you gain electrical access to that neuron to record its activity in response to different drugs and even different levels of oxygen, which I was studying. And there's so many variables need to be controlled and just perfect.
So many things can go wrong.
Yeah. I remember we were using glass electrodes, and we switched the different glass electrodes, and it come to find out the glass was coming from like a different quarry or something. And the electrical properties of that glass prevented me from getting a gigaohm seal on the membrane. So it was some variable that I could have never even predicted why my experiments weren't working. But yeah, I did 2 years of really hard work without getting any data. So I think the biggest thing from my perspective is persistence. I think persistence will pay off, but to also ask for help. I was always kind of hard-headed and wanting to do things completely on my own, but really had to more or less maybe humble myself and just start asking people for help. Like, this is just not working. I can't do this. And that was kind of a new thing to me, to just keep asking people for help until I was able to get enough information and feedback to modify my approach to get the data that I needed. But it took me about 2 years to do that, to get any data at all.
That's amazing. I think I would have given up by then. Well, Dominic, I think you brought up two traits that are critical for scientists, or maybe critical for scientists to learn as they're going along their journey. And that is, as you mentioned, persistence, and also just being resourceful, not only in asking for help along the way, working with your colleagues to get the information you need, but being resourceful in terms of funding, being able to diversify your funding portfolio. And I think the more we progress in this difficult funding environment, the more we're going to see these collaborations between industry and government contracts and academia.
Yeah, it's really, the government, I just saw the NIH funding is going to be cut pretty significantly, like 20%. And it's like half of what it was when I was a graduate student 20 years ago or more. So I have concerns over my Ph.D. students. If they want to stay in academia, they have to be resourceful and partner with industry. And I think you need a diversified portfolio. It's like if you're going to be investing, right? You don't want to put all your eggs in one basket into the NIH. I always kind of prioritize the NIH because it's like the top level peer-reviewed from a grant perspective funding, but there's many other funding sources out there. We're going for an NSF grant now, but Department of Defense and NASA and many different foundations too have helped us start up pilot projects that then allowed us to collect the data that we can put towards bigger projects. And I think it's been really important for us to reach out, or for foundations even reach out to us because they see the kind of research that we're doing, to go for smaller grants. And that data collected becomes seed data for larger grants. So really important for me to do that.
Absolutely. Well, thank you so much for sharing your challenges and how you worked through them with our listeners today. But we don't just want to focus on your tough times, Dom. Can you share with us next one of these success stories, a happy moment in science that meant a lot to you?
Yeah, I think the things that make me most happy is seeing the people under me get success. And I have been very blessed to have amazing people in my lab. So I have 2 presidential fellowship students that are really typically, historically, have been hard to get, especially for our department. So they just happen to want to work in the lab. And just this week, I got an email from the American Physiology Society, APS, and my undergraduate student, Janine DeBlasi, and she's honors undergraduate student, and really she's the hardest working student that I've ever seen. And we worked on her fellowship together in writing up support letter, and she had some fantastic data that we've collected in the lab, and she was able to get American Physiology Society summer fellowship, and she just got 2 awards on top of that. So that was probably, over the last couple of months, that's like the biggest win for me, because that fellowship also brings money into my lab to support her and to support all the science behind this project, and also gives money to allow her and my students to attend scientific conferences to present this data and disseminate the data and connect with other scientists and network. So that's one of the recent success stories really proud about for all the students working under me. They work incredibly hard, and they're super smart, far smarter than I was as an undergrad or graduate student at their stage. And not all students are like that. So I have to emphasize that the students that have come into my lab are really, really out-of-the-box thinkers and exceptionally talented and smart. So that's been a huge win for me, and a big factor in me actually being able to do the science and be productive enough to get tenure at my university, is having great people under you that are collecting the data that results in publications and also allows us to be competitive for other grants.
Absolutely. Well, congratulations to you and your students. It sounds like you've got a wonderful group there.
Yeah, I'm super lucky and thankful every day for that.
Definitely. Well, thanks so much for sharing one of these success stories. And I think there are so many different kinds of successes in science, but it's fun to escape the scientific world for a little while. We talked about some of the things you like to do when you're not doing science. And one of the things I love to do is pick up a good book and enjoy some non-scientific reading. So I'm going to quiz you today, Dom, for a book that you've read and really enjoyed. Is there one that you can recommend to me and our listeners? And it can be a science book or a non-science book.
I love to immerse myself in books too. It's a great kind of escape from the hectic goings on of normal life. Yeah, some of the books that come to mind, I actually listen to a lot of audiobooks. And when I'm traveling or driving, I do that. And if it's really good, I'll end up buying the book too. But Tripping Over the Truth by Travis Christofferson was an excellent book. And I read it and I was so impressed by it. I reached out to Travis, and actually I was able to write a foreword for his other book, or the newer version of it. So Tripping Over the Truth is the original one. I bought a lot of copies and gifted to quite a few people. Another book I recently read, or should say listened to, was Ego is the Enemy by Ryan Holiday, which is really an excellent book. I mean, it's just kind of focuses on like stoicism, and it's more of a philosophy kind of book. And I guess the third book that almost 3/4 of the way through is a book called Man on the Moon. And that's a fantastic historical story about the Apollo missions, but it's told in a way that's much more engaging than other books that I've read. And of course, because we're doing some work with NASA, I like to kind of educate myself as much as possible on the history and the people that were involved in making spaceflight possible. There are 3 vastly different books, Tripping Over the Truth, Ego is the Enemy, and Man on the Moon, but I like to delve into different subjects, kind of escape from the everydays going on in the lab.
I'm the same way. I pick up any old book. I'm not always drawn to a particular genre or fiction or nonfiction. I'm all over the board. So Great recommendations. We'll put them on our website for our listeners to find there if they are ready for their next book. And we chatted a little bit about some of the places that science has taken you over the course of your career. But if you had to think back and pick one favorite, Dom, where would that favorite place be?
I would have to say New Zealand. I think it was 2.5 years or 3 years into my Ph.D. program, and my advisor asked me to give a talk in New Zealand. And it was 2 presentations in Auckland, Christchurch, and Rotorua. So it was like 3 different locations. And at the time, I had never been outside of the United States. So to be on the other side of the planet, and I think they're like 17 hours ahead or something like that, it was such a real experience to be asked. It was sort of a last-minute thing. And I gave a presentation to the Neural Control of Breathing group for Physiological Society. And that was an experience of a lifetime. And that wouldn't have happened if I was not in science and doing science. And it allowed for a fantastic trip and a terrific opportunity for me to share my science and connect with people.
Definitely. I think some of these travel opportunities are amazing because you get to connect with scientists all over the world who share some of your interests, but then also getting to see these different places. Were you able to do some sightseeing while you were over there?
I did, yeah. New Zealand is an amazing place to visit. There's such a diversity in environments, from rainforest to skiing in the mountains you could do in the same day. You could be swimming in the water.
And then down to the sulfur springs in Rotorua, yeah.
Yeah, and Rotorua is kind of like this really strange place. You get off the plane and you smell this egg sulfur. But it's an amazing place to visit, and I was really lucky that my science was able to take me there, and really to dozens of places over the years. But that was the first really exotic trip that I took as a scientist, and I was like, I could get used to this.
Right.
And it's so great to share it too with your colleagues. So we took a variety of tours through New Zealand, and it's great to experience these places with the scientists that you're friends with and that you have this sense of camaraderie with. It's a great experience to share.
Definitely. And speaking of this camaraderie, we spoke earlier about how wonderful the group you have working in your laboratory is. And I think some of the people you get to work with in science are part of what makes it so wonderful. And I know there's lots of different personalities in science, and there are some stereotypes out there about what scientists are like. And we try and break those on our show because I think there are so many different types of people in science. So do you have a story of yours, Dom, that you can share that highlights the human side of science? Maybe these funny traditions or quirky experiences that you've had with your colleagues?
Yeah, we've had a variety of characters that I've worked with throughout the years through my postdoc career and even as a junior faculty member. And I was working with one postdoc, he knew I had a fear of spiders. So when he would find a spider, one of our labs was actually like in the basement of this place when I was working in Ohio and even in New Jersey at the time. And he would collect a spider and put it in a place where if he knew I was going to move my book or something, the spider would jump out.
Oh, that's cruel.
So I worked with a group of people who they love to shock people because I had a startle reflex. I loved quiet time and just working alone. But at the same time, when someone would come up to me, I would have this over-exaggerated startle reflex, which is also a factor when we were working with hyperbaric chambers. This week, actually, my colleague, and he used to be my postdoctoral fellowship advisor, so we work on hyperbaric chambers, and he bought a train whistle from like the 1800s and plumbed it into the chamber. So when the chamber was pressurized, he could release some of the pressure by pulling on the chain of this train whistle, and it just shocked us all down the hallway. You could hear it. Yeah, it's really fun to be working with people who don't take themselves too seriously and can periodically do things to just totally get you laughing and to even shock you out of what can sometimes be a mundane existence when you have to do a lot of committee work or paperwork and things like that. But generally speaking, my life as a scientist is extremely exciting, but you do have a lot of tasks and paperwork associated with academia, and it's good to break that once in a while with laughter and shocking people by doing crazy things.
Right. Well, and I've been impressed too with the people I work with, how creative and funny scientists are, because I don't think we have that reputation, but so many places have this culture of pranks, practical jokes, and working hard, but having fun at the same time.
I think you need to, because otherwise you're just going to go crazy.
Right.
Scientists have this kind of insatiable work ethic because if they're successful, they're like so passionate about what they do. And I know my wife in particular, she really has to drag me away from things or else I'll just immerse myself in the weekends. And you got to budget in time to get away from it all, ideally out into nature. And that's sort of our downtime. So, and just having fun and laughing and doing crazy things is a way to break it up.
And I think, speaking of being passionate about it, I think a lot of scientists get really excited about the questions that they get to ask in their work, so they don't want to stop. Dom, if I gave you everything, if you had all the resources, funding, technology, staff, whatever you needed in the world, what is the question that you are just dying to know the answer to?
My Ph.D. student asked me that last night when we were driving home.
Really?
Talk. It kind of had me stumped because I turned the question back on him and he couldn't answer because there's so many things. But if I really think about it, I think the main thing is our cancer research program has unexpectedly kind of took hold after we made some observations a few years back. And I have half of the lab working on cancer research projects. And one of the big questions is what types of cancers are most vulnerable to the metabolic-based therapies that we're developing. And we're looking at the ketogenic diet, ketone supplementation, but we're also looking at cancer-specific metabolic drugs, common things like metformin and other drugs that can target sugar metabolism. So if I had unlimited funding, staff, and technology, what I would want to do is get cell lines of all the different human cancers that are most deadly, at least the top 20, and to vet out and determine what cancers are selectively vulnerable to the types of therapies that we're developing and testing, and to do that in cell lines and then ultimately move that to animal models to further validate those approaches, because we study mostly brain cancer. And a lot of questions are, will these therapies also work for other types of cancers? Or even a particular type of brain cancer, whether it's a glioblastoma, that brain cancer can be different depending on the person too, and it can mutate in various forms. So that would be kind of the main project that I would put my energy into, determining what cancers are most amenable to being treated with the therapies that we're working on.
Well, it sounds like you've got the rest of your lifetime research project all sorted out then, Dom.
Yeah. So you start in a way where you can do kind of a high yield testing of this and determining through cell-based assays what's a low-hanging fruit, and then work from there. Another project, I know you just asked for one, would be a longevity study. So we're doing some projects now, and we have mice that are aging and probably equivalent to like a 50, 60-year-old human. If we put them on various regimens, if we give them calorie restriction, and then we compare that to the ketogenic diet, and then we compare that to ketone supplementation, and then we compare that to metformin, and we let those mice live a happy, healthy life, how long will they live? Will extend their life? And at the end of their life, if we know that 20 or 30% of them typically get tumors, but we find that only 5% do, that's a significant finding. So that's sort of the studies that we're doing now, but we're limited by our funding because we have to pay like cage charges every day for animals. And you need a lot of mice to do this. And it's kind of interesting because you're not doing any intervention. You're just changing their diet and maybe incorporating some of the low or non-toxic drugs into their diet, but just letting them live their healthy life and determining if their lifespan is. There's not a whole lot of people doing prevention studies or longevity studies. And I think it's really needed. And so I think directing funding into those kinds of studies is something that's a close second, but it's very close to my interest in the cancer study that I mentioned.
Well, I'm intrigued. Are there any hints of what kind of diet might be most conducive for longevity? I know I've heard great things about things like restricted calorie diets, but how does that compete against the others? Do you have any thoughts at the moment?
I kind of do. And what I want to test is, I guess you would call it supplemented ketogenic intermittent fasting. So the human equivalent, I guess, you would be eating between, say, 4 p.m. and 10 p.m. every day. And in that window of period that you're fasting, you do exercise ideally. And we can mimic that in mice. So just putting a running wheel in there, and they'll run without even forcing them. So to do kind of fasting and exercise during I guess it would be like 18 hours of time that they're fasting, and also have access to ketone supplementation, which we know when consumed orally in small amounts does not increase insulin. So you'd have intermittent fasting stimulates autophagy, especially if you couple it with exercise, and keeps factors like IGF-1 and other things that are associated with longevity. It can activate AMP kinase, stimulate autophagy, suppress mTOR, and then within the window of eating to have a very carefully formulated ketogenic diet with specific macronutrient ratios that would not stimulate these kind of hormones or excess growth, and even allow the animal to eat just enough to sustain their growth and repair, but not to stimulate consumption of surplus calories. So if it was a high carb, sugary, salty diet, they would tend to consume a surplus amount of calories and kind of negate some of the effects that would be derived from intermittent fasting. But if during that eating window, they were eating low carb, I think. And I think we are much more receptive to this kind of intervention the later on in life. I think if we're younger, like I can reflect back, I could eat massive amounts of sugar and carbs and be perfectly fine, and my blood work looked fine. But as we age, our carbohydrate tolerance decreases with age. So this may be something to implement kind of later on in life as a longevity strategy, in my opinion. And that's kind of how we're going to test it.
Excellent. Well, Dom, thanks so much for sharing some of these big questions on your mind. And I know we've talked a little bit about your research over the course of our conversation today, and also information about your career path. And I love sharing with our listeners any advice that our guests can give about their own career paths. So is there one piece of advice that you received at some point that really helped you that you can pass on to our listeners today?
I think it's to be really flexible in your belief system, whatever that is, even philosophical and even from a science perspective. I've approached different projects with the idea of this is what we want to test with our hypothesis. And I kind of maybe harped on this a little bit, and not really being flexible with adapting that model system to the data or other people's findings, and almost being kind of not humble. There was a quote that said, when you get to the point where you think you're humble, it means for certain that you're not humble. So sometimes the research that we do, we're so convinced by some of the studies that we've done that a particular approach is the best approach, but it may only apply to a certain scenario. So I think it's really important to be flexible with your beliefs. And if your own data proves you wrong, to go back and say, I was wrong on this. For example, the ketogenic diet maybe doesn't work for this type of cancer or that type of cancer where we thought that it did in the past. But as we did more studies and more data, as the data emerged, it's not the best choice for this or that type of pathology. So I think it's really important as scientists to really seek the truth and to let no kind of bias prevent us from seeking the truth and also disseminating that in our literature. I think it's really important.
I definitely agree. I think keeping that open mind often helps you avoid those biases. If you're really not sure how it's going to turn out when you're going into it, then it kind of helps you be more objective and see the truth that's in front of you.
Absolutely.
Great advice to leave our listeners with today. Is there any other last piece of advice, Dom, or a last note of inspiration you want to give them?
Well, I guess coming from that perspective is to seek mentors that are very flexible in their thinking and humble in different ways and that have integrity. So I really want to surround myself with people that are not necessarily the most prolific scientists or the most successful, but the ones that I feel have the highest integrity in doing what they're doing. And I think it's really important for young scientists out there, depending on where they want to go, to identify several people who have achieved specifically what they want to achieve and try to get close with that person and try to get that person as a mentor, because it's really the only way that you're going to advance your career in the most efficient way possible, is to emulate people that have done what you're seeking to do and to figure out how they achieve that. And you may pick 2 or 3 or 4 people and they all chose a different path, but you can garner information from each one of them in ways that they did that. And I think and looking back, I think that was really important for me to surround myself as much as possible with people that are already at a position where I want to go. And many of them did it in totally different ways. And that becomes probably the most important piece of advice.
Absolutely excellent advice to leave our listeners with. Listeners, choose your mentors wisely and strategically. I think you kind of hit on both of those points there, Dom. So thank you so much for joining us today. Can you tell our listeners, if they want to learn more about you and your work, where should they go?
I have a website that I compile information on for people that are interested in, for example, the ketogenic diet or metabolic-based therapies, or even looking for like a consultant. It's called Keto Nutrition, all one word, .org. And just recently, we kind of started a company called KetoneTechnologies.com. And it's more of a research and development company. And we'll be doing consulting and maybe public speaking, but also we're working on acquiring the licenses for various patents. We'd work with different individuals or companies that would want to develop these patents into products. So Ketone Technologies recently formed this week, and KetoNutrition.org and KetoneTechnologies.com are the 2 sites I direct people to.
Excellent. Well, listeners, definitely check out these websites. And Dom, thank you so much again for joining us on the show. It has been a pleasure to chat with you.
Thank you for the opportunity. I really appreciate it.
Well, it's been wonderful to hear more about your story. And listeners, thank you for joining us as well. We'll see you next time on another episode of People Behind the Science. Your voyage to explore the lives of today's exceptional scientists has just begun. You can find everything we talked about today, including our guests' favorite books, biographies, photos, and more, when you visit us at www.peoplebehindthescience.com. I look forward to chatting with you next time on People Behind the Science.